Good morning, students. In a previous video, we have seen that the nitrification, and the ammonification. In this video, we are going to see reductive amination and the transamination. Okay, let's welcome. Let's go. Let's start. The atmospheric free molecular nitrogen it is converted into fixed NO3, NO2, and the NH3 are absorbed by the plant. But the high level of ammonia in a soil around the root zone is toxic to the plant. Hence, the plant avoid the ammonium toxicity, rapidly converting ammonia into useful amino acid is involved in a protein synthesis. The conversion of ammonia into different amino acids such as glutamine, glutamate, asparatate, asparagine plant is take place in two major processes like the reductive amination and the transamination. Reductive amination is the process which ammonia is assimilated into plant by converting into amino acid like glutamate through the process of a reduction process. The reaction involved in this process. Let's see. Alpha ketoglutarate plus NH3 in presence of NADH it gives NADP GTH. Glutamate is converted plus H2O is a form that is known as the reductive amination. The alpha ketoglutarate is produced from the OAA in a TCA cycle. OAA that means oxaloacetic acid. It is combined with ammonia in presence of the GDH glutamate dehydrogenase. In during reduction NADPH is converted into NADPH. The enzyme GDS is located in the chloroplast and the mitochondria. The glutamate assimilated into plants even it converted into amides. It is known as a glutamine. In this reaction, glutamate is combined with the ammonia, producing the glutamine in presence of the enzyme glutamine synthetase. In this reaction, 1 ATP is used, which converted into ADP plus PI enzyme. GS is located in a chloroplast and the cytoplasm of a leaves and the root. In this process, ammonia is react with the alpha ketoglutarate acid to form the glutamic acid by the reduction process. Hence, it is known as the reductive amination. The reaction are given in figure. That is overall about the reductive amination. Reductive amination, it is nothing but the reduction of ammonia into amino acid like the glutamate in presence of the GDS. NADH is converted into NADP. That are the over in the reduction process, reduction amination. Next, transamination. The glutamic acid is the main amino acid from which 17 amino acid are from. That is the important concept or the important sentence. Glutamate it is a important amino acid from which 17 different amino acid are the form. Through the transamination, each amino acid made up of the one carboxyl group and the one or more amino groups. Okay, is that clear? In that first amino acid are the different amino acid are formed by the glutamic acid. Okay, in each amino acid there is a one carboxyl group and the more than more amino group are present. Transamination involves transfer of a amino group into one amino acid to keto group of a keto acid, and the enzyme are responsible for the transamination, that is transaminase. The different amino acids are formed from the ammonia by the transamination process are absorbed by the plant. Once assimilated into glutamine and the glutamate, nitrogen is incorporated into the amino acid via transamination reaction. The enzyme catalyzes the reaction that is known as amino transferase. An example is aspartate transferase. which catalyze the following reaction: glutamate, oxalo, oxaloacetate, aspartate plus 2-oxaloaspartate. In the above reaction, the amino group of glutamate is transferred into carboxyl atoms aspartate. That is aspartate. It is an amino acid that participate in the malate aspartate shuttle to the transfer reduction equivalent from the mitochondria, chloroplast into cytosol, and in the transport of a carbon from the mesophyll to the bundle sheath for the C4 carbon fixation. All transamination reactions require the pyridoxal phosphatase as a cofactor. Let's say glutamine plus aspartate plus ATP, it gives the aspargine, glutamate, ADP plus IP. The nitrogen from the glutamate is transferred from the aspargine in presence of enzyme aspargine synthase. The transfer of ammonia from glutamate to the aspartate and the aspargine by the transamination reaction. That is the two concepts I have explained. First, that is a reductive amination. That means conversion of ammonia into different forms of amino acid, that is glutamate or the glutamine. In this figure, I have explained that is atmospheric nitrogen are present. Atmospheric nitrogen are fixed by the different organism or the non-organism mix factors into ammonia. In the plants, there is more amount of ammonia is formed, due to which toxicity of ammonia will be take place. To avoid that toxicity in a plant, transamination and the reductive ammonation will be take place. For that purpose, some process are the run in a plant. In that first, reductive amination. How reductive amination will be take place? Let's see. The ammonia is converting into different enzyme. Okay, ammonia are converting to different amino acid, and that are helpful for the plant. Especially amino acid, in a amino acid is a reduction will be take place in presence of a NADPH, NADPH and the GDH. Okay, let's see what are that. Alpha ketoglutarate plus NH3 in presence of NADPH, it gives to NADP to form a glutamate, and it gives the H2, and glutamate it is the important amino acid. And after the glutamate are formed, again ammonia are binds with the glutamate in presence of a GS, that is a glutamate dehydrogenase. It gives to the glutamine amino acid. And transamination, it is a simple process. Transamination, that is conversion of a glutamine or glutamate into aspartate enzyme or the aspartate amino acid. That means transferring the amino group from the one amino acid to the another amino acid in presence of the enzyme transaminase. The enzyme catalyzed by the amino transferase. For example, aspartate is catalyzed by the following reaction: glutamate oxaloacetate plus oxaloglutarate, that is glutamine aspartate, ATP, aspargine and glutamate. It gives ADP plus PI, that is transferring one amino group from the glutamate or the glutamine to the aspartate, and from the aspartate it again transferred with the aspargine. That is overall about the reductive ammonation and the transamination.